Um, our next speaker is John Watts. John joined the RNA Therapeutics Institute at UMass Medical School in 2016. Before that, he was an assistant professor at the University of Southampton in the Department of Chemical Biology. In 2013, John was the recipient of the Oligonucleotide Therapeutics Society Young Investigator Award. And today he's going to tell us about his work using RNA to fight COVID-19 and RNA virus. John, thank you.
Well, thanks very much. This is a really fun seminar series. I think, in a sense, this collaborative seminar series between the different RNA institutes is one of the really very good things to come out of being locked down. So thank you all for joining us today and for the talks that you've been giving on other weeks. Today, I want to give a talk about some of the work we've been doing on translational research in the lung, and in particular, what we've been doing during this pandemic to see if our technology can make a difference. This has been a very collaborative project. It's also still very much a work in progress. This has been mainly led by my group and by Anastasia Kurova's lab. Our viral live virus work has been done in collaboration with Jennifer Wang and Bob Finberg's lab, except the in vivo, upcoming in vivo viral work, which is done by Anthony Griffiths at Boston University. Sometimes rapid drug development is particularly pressing. We all want to develop drugs quickly, but in the case of a pandemic, we feel this more acutely than usual. So 100,000 people were diagnosed with COVID yesterday. It's been around 100,000 every day since the beginning of April, essentially. Over 8 million people have been confirmed positive, over 440,000 have died. And so it is important for us to identify existing drugs and develop new drugs as rapidly as possible. One of the really exciting things about RNA therapeutics is that we can separately optimize platform technologies from particular sequences. And that means platform technologies, as you can see here, for oligonucleotide drugs are mainly dictated, the pharmacokinetic and pharmacodynamic properties are mainly dictated by the chemistry of the backbone of the oligo, or the vehicle in which it's delivered if it's a formulation, or the ligand if it's a conjugate. And in contrast, the sequence of the bases in that drug are what will determine the target recognition and modulation. And so this ability to separate platform technologies' PK/PD properties from particular disease targets is one of the things that differentiates oligo drugs or other nucleic acid drugs from traditional small molecule drugs, where the chemical structure is the thing that determines both the target recognition and modulation, as well as the PK/PD absorption, distribution, metabolism, and excretion properties. So if a platform technology is established, we can do rapid drug development. If we know how to modify the bases or the ligand or other aspects of a drug to deliver functionally and robustly to a particular tissue, to have it last long enough to produce a good therapeutic effect, and so on. If all of that's in place, we can do really rapid drug development by changing the sequence of bases. So for example, Moderna, within 2 days of receiving the sequence of the SARS-CoV-2 virus, identified their clinical compound for their vaccine. They already knew how to make RNA vaccines, so they think this is, of course, now their front-runner program. But being able to move from a sequence to a drug very rapidly is a hallmark of nucleic acid therapeutics. But of course, all of this depends on having an established platform technology, and to develop an established platform technology is a much longer process involving new chemical modifications, of course, in vivo proof of concept studies, iterative optimization to identify what has worked and what hasn't, and ultimately, before we can really say we've got a validated platform technology, we need the evidence that it works in the clinic. And in terms of gene silencing in the lung, that is not where we are at the moment. We've been working on platform technology for the lung, but there's nothing that's really been, gone all the way to FDA approval, for example. in terms of ability to robustly silence different genes in the lung. So today I want to tell you about both of these aspects. First of all, this Diana-4 platform technology. My lab, as well as Anastasia's lab, have been working on platform technology for the lung for a couple of years before the COVID pandemic. But for obvious reasons, we've put a lot more focus on this work in the past 3 months. Then in terms of the Pharmacophore, the specific applications in this disease, I'll tell you about some work that we've done to identify sequences that inhibit the novel coronavirus, as well as relevant host factors, and then tell you where we are in terms of therapeutic development. So we could in principle deliver oligonucleotides to the lung either through the circulation by systemic administration or by local delivery to the airway itself. Systemically, there are advantages in terms of ease of clinical delivery through a simple injection, uniform lung distribution through capillaries, no impact on lung inflammation or other aspects of patient's clinical condition in the context of a respiratory virus, and something that kind of cuts both ways, significant exposure of other organs. So that might have liabilities in terms of off-target toxicities, but also in a disease like COVID, where there is a significant amount of systemic pathology, there might be some advantages to exposing other organs to the drug. On the other hand, a locally delivered drug is less invasive than an injection if it can be delivered by nebulization. It maximizes the extent of delivery to the respiratory tract. However, it may be affected by the patient's clinical stage in a respiratory virus. And again, this cuts both ways. It's highly selective. We don't get much exposure to other tissues, which may have advantages and disadvantages. So Anastasia's lab identified that specific lipid conjugates drive uptake into the lung of fully modified siRNAs. And so you can see this data from Annabelle Biscan's paper published last year, showing either an unsaturated, polyunsaturated fatty acid EPA, or two variations of a fully saturated fatty acid conjugate drive a significant amount of uptake into the lung after either sub-Q or IV delivery. My lab focused on a slightly different approach, doing local delivery of antisense oligonucleotides. So we've observed that one of the real crucial modifications is the nature of the sugar modification. So these are gapmer antisense oligos. They have wings on the 5' and 3' end of high affinity sugar modified nucleotides that ensure tight binding to their target RNA. And then those wings flank a gap, we call it, of DNA, which allows recruitment of RNase H to cleave target RNA. In some tissues, we love Mo gapmers. We find, for example, in the brain, we use a lot of Mo. In the lung, we don't find that Mo gives us the potency that we need. And so we focus our work on LNA gapmers, as you can see at this graph on the left. We see a significantly larger extent of silencing by LNA gapmers relative to Mo gapmers in the lung. There's a sort of tissue-specific aspect of medicinal chemistry here. The lung, of course, is very complex and has a number of highly specialized tissue types. Epithelia that are exposed to air, endothelia exposed to circulation, structural cells like fibroblasts, and immune cells of a variety of types. So my postdoc, Minwoo Shen, developed a way to measure, sort of easily measure silencing in different cell types by measuring knockdown of a ubiquitously expressed cell surface marker called CD47, which is a don't eat me signal. Knocking down CD47 allows us to then sort the cell types into their different populations using flow cytometry, and simultaneously in the same flow experiment identify the extent of silencing. Now, it's interesting to notice that although the first cells to contact an intratracheally delivered oligonucleotide are obviously the airway epithelia, those are not the cells that show the most robust gene silencing. In fact, the cells that show the most robust gene silencing are fibroblasts of the lung. And you'll see this coming back a number of times. Immune cells, while they take up a lot of oligo in a kind of gross manner, don't show a lot of functional silencing. again, that's sort of consistent with their role of being able to internalize things and quarantine them, prevent them from doing much, at least initially. And we've begun to apply this LNA gapmer approach to silence disease-relevant genes. In particular, this gene ADAM33 is an asthma susceptibility gene that's selectively expressed in fibroblasts. And so the fact that we can silence it, this is out to 3 weeks after a single dose, the fact that we can silence this is additional evidence that we're getting effective silencing in lung fibroblasts. So when this pandemic hit, members of our labs, my lab and Anastasia's lab, began really taking a lot of initiative to suggest that we apply what we have been doing in terms of platform technologies to the lung, for the lung, to explore whether we could further improve those technologies and develop sequences specific to the virus. And since our labs were closed for most other purposes, instead of being spread way too thin like we all are as academics, we put essentially all of our energy for the past 2.5 months, at least all of the actual wet lab energy, into improving platform technologies for the lung and identifying sequences for the virus. So the remaining data here is new data on new conjugates and designs, exploring whether we can identify better ways to silence gene expression in the lung. So these are with fluorescently labeled oligos. These are different conjugates. This is here in the left middle, an siRNA that's not conjugated. When we deliver it to the lung, we see some uptake in the lung, but relatively little compared to this optimized local conjugate. That works differently than these two conjugates shown on the bottom right, which are delivering the material to the lung after systemic administration. You can see that the higher extent of silencing by conjugate 1 systemically and conjugate 3 locally correlate also to a higher extent of functional silencing in the lung by those same designs. So we were able to use Minwoo's CD47 cell type specific assay to explore some novel conjugates and their ability to silence gene expression, again, in different cell pools. Each of these sets contains first 2 negative controls, saline and then a non-targeted control. Finally, then an unconjugated siRNA, the third one is unconjugated, and then 3 conjugates. We'll call it A, B, and C. So you can see that, again, in fibroblasts, we're able to get good silencing even with an unconjugated asymmetric siRNA, whereas in endothelia and epithelia, these conjugates significantly improve the extent of silencing. We have another new recent scaffold that shows significantly improved silencing. CD47 and some of the other targets in the lung, we have had a hard time getting silencing to go much below 50% in many of these cell types for some targets, unlike the antisense oligo targets that I showed you in the first part of the talk. But you can see this newer conjugate allows more robust silencing in fibroblasts. It also works well in endothelia. So this is now, I should say, 2 sequences using the same conjugate, conjugate D. However, airway epithelia are still showing relatively poor silencing at this time point. So we're in the middle of carrying out another kind of pivotal scaffold comparison and optimization study, comparing a number of systemic conjugates, 2 systemic and 2 local, and carrying those out to extended times. Some of our earlier work suggested that a week might not be long enough to see maximal silencing. And certainly we would expect a pretty good duration of effect out to at least a month, possibly significantly longer. So I want to switch gears a little bit. While we're working on these platform technologies, in parallel, we've also been focused on identifying sequences that inhibit the SARS-CoV-2 genome, RNA genome, as well as relevant host factors involved in the disease. And so the goal would be to deliver a cocktail of oligonucleotides, siRNAs or antisense oligos that could inhibit either host factors or the RNA genome, or a combination of those, either as a treatment or as a prophylactic, given the long duration of extent. So this is a schematic of the viral genome. It's about 30 kilobases long. And a PhD student in Anastasia's lab, Katie Monopoly, computationally identified a number of sequences to all of the viral genes. So overall, between the different viral factors and host genes I'll be telling you about, we've screened over 500 oligos in the past few months. So you can see the viral genome contains 2 long polyproteins as well as some specific factors, spike, nucleocapsid, and so on. So we identified, Kitty identified regions that were as highly conserved as possible in all of the patient isolates that were available at the time. That was 700 and something patient isolates. And you can see how the distribution of siRNAs compares. So these are selected, predicted to be active, as well as with a weighting for the most highly conserved regions of the virus. To screen these, we introduced the sequences, the target sequences, into a SciCheck reporter system, which allowed rapid screening of as many targets as possible. You can see that we were able to identify siRNAs that have activity against this reporter for targets in essentially all of the viral genes that we looked for, some better than others, of course. In parallel, we screened antisense oligos against those same conserved regions of the SARS-CoV-2 genome. And again, we're able to identify robust hits against those different regions. So besides the targeting the viral genome, we were able to identify sequences against ACE2 and furin. Both of those are factors that are relevant to viral entry into cells, as well as these are, here I'm showing dose responses on a subset of the compounds. We also have sequences against other factors, including innate immune factors involved in disease progression, disease morbidity, like to minimize things like cytokine storms. So reporter assays are all well and good, but do these actually inhibit the live virus? So our initial experiment to explore the live virus has been what we call a cytopathic effect assay. Again, this was done in collaboration with Bob Finberg and Jennifer Wong's lab. And in parallel, we have sent a set of our compounds to the NIH to be tested in CPE assays in parallel with other compounds they're working with there. So this challenges the cells, cultured cells, with virus at low MOI, and then looks over a period of some days to see whether cells are showing evidence of malformation or syncytium formation. So this is a sample of 2 of the plates. We've worked with a dozen or so plates now, set up a dozen or so plates of CPE assays through the hard work of these 4 people listed at the bottom right. And we, as you can see, have identified a number of sequences that seem to show a protective effect. The green wells indicate that no CPE was seen, so the cells look healthy. In contrast, the wells that are red represent compounds that were not active. And so each compound was done in triplicate. You can see these represent 3 copies of the same well. In some cases, we see all 3 showing the consistent behavior. In others, it's less consistent. Of course, this is a digital assay, and so we're now carrying on with additional assays like qPCR for viral genome copies and immunofluorescence for viral proteins, because of course inhibition of the virus is not only a digital effect. We need to also identify the extent of silencing of viral RNA and so on. So that set of experiments is also underway. So, so far, I've told you about some summary with, some progress on both platform technology for the lung and sequence selection for SARS-CoV-2 transcripts. So far we see over 4 weeks of silencing in the lung after a single intratracheal dose of antisense oligo. The newer designs that we've worked on over the past couple of months are likely to be longer, but those experiments are still underway, so we don't know how long they'll last yet. I think one thing that you may have noticed looking at these data today is that this rapid progress that we've made in these newer platform technologies, which was motivated by the COVID pandemic, we hope it will be useful for COVID, but it may be even more useful for other lung diseases. And in particular, while epithelia and endothelia are very important for COVID, fibroblasts within the lung appear to be particularly susceptible to functional uptake of antisense oligos and siRNAs. And fibroblasts within the lung are responsible for driving, or pathology within fibroblasts, let's say, is responsible for driving a number of other significant clinical unmet needs. So we're following up on applications within things like pulmonary fibrosis, for example. Where are we in terms of COVID? Well, we're finalizing our selection of both the sequence and the scaffold at the moment in parallel experiments. We will then put those together, assemble cocktails of 3 sequences to minimize the possibility of viral escape. We have in vivo antiviral efficacy studies booked for July with Anthony Griffith's lab at BU. And if successful, the timing of that study would read out in time for us to initiate a first-in-human emergency use study as early as September. So are oligo drugs able to respond to a pandemic quickly, I think is an open question still, especially when there's platform technologies still to be done. We know there are other companies also working on this in parallel, and we're delighted for their progress as well. But we certainly hope that we will continue to identify sufficient progress to see a real difference in viral progression in vivo. I'll end by putting back up the acknowledgement slide again, because I just again want to underline what a team effort this has been to move forward this project. It feels like it has moved very quickly for a drug development project, although it still feels painfully slow in the context of this pandemic. So I'll stop there. Thank you for your attention.
John, thank you so much. We're all hoping that this works quickly and effectively. Sadaf asks, can you hypothesize why epithelial cells are more difficult to silence?
No, that's something that we think about a lot. Clearly, they must be being exposed to the oligonucleotides when they're delivered locally. So if those of you listening, if you have ideas as to what could be happening in terms of trafficking, we welcome your ideas. I think about what this might mean in terms of trafficking, but I don't have an intelligent enough answer to propose more than hand-waving answers as to what could be happening trafficking-wise to allow better functional uptake in cells that are deeper into the tissue.
Shunqing Liang asks, how did you quantify the slicing percentage for different cell types in the lung after delivery?
So there's a laborious way to measure silencing in different cell populations, a number of laborious ways. For example, we've done FACS and sorted out cell populations and then measured silencing by qPCR. We've done single cell sequencing. I didn't show you either of those approaches today, but they give consistent data for antisense oligos. The way that my postdoc Min Wu has identified that allows us to make much quicker progress is that CD47 assay. So with a single flow cytometry experiment, we can sort the cells into their populations and measure the extent of CD47 remaining on the surface of the cells, which have been stained with a CD47 antibody. And so that allows us then, with one injection, to measure silencing in particular cell populations.
Lucas Philippe asks, nice talk. How did you typically look for off targets in such a short window period?
Yeah, so great question. So we haven't done things like sequencing, whole, transcriptome-wide sequencing to explore off-target silencing at this point. As we are able to identify leads, we will be pushing those through a more demanding set of safety studies. So far, we've ruled out things that have obvious toxicity in cell culture, but that's as far as we've gone so far. The best way to do that experiment is to use something like a next-gen sequencing run to measure changes in transcripts, transcriptome-wide, of course.
Katie Mouzakis, who's a PI at Loyola Marymount, asks, do any of your siRNAs target the SARS-CoV-2 frameshift site RNA?
That's a great question. Not that I know of. I would have to ask Katie to be sure, but I don't believe so.
Because in theory, right, you wouldn't have to use a gapmer if it simply blocked frame shifting.
Yeah, yep, that's a great idea.
Payao asks, will the same strategy work effectively in fibroblasts from other organs such as heart, like cardiac fibroblasts?
So I haven't done the cell type specific experiment in heart yet. With some compounds, including LNA gapmers, we see some silencing in heart. I know some of Anastasia's lipid conjugates give silencing in heart, but I don't know what the breakdown is between extent of silencing in cardiomyocytes versus fibroblasts, for example. That's a really interesting question. Thanks for that.
It's now 5:01, so if you could answer the remaining 4 questions through the chat window, that would be terrific.
Absolutely.
Thank you all for attending today's Zoom seminar series, and we look forward to seeing you next time. Bye.
Thanks.